Hello all, welcome to F.Y.B.Sc class. Today we are going to discuss about the design and preparation of microbiology media from the unit Microbial Cultivation and Growth of F.Y.B.Sc course MB122 Microbial Cultivation and Growth. The learning objectives of today's lecture is to study constituent of nutrient media, what are peptone, what is the role of peptone in nutrient media, what why the water is commonly used as a dissolvent, what are yeast extract, what are yeast extract, how we can obtain them, what is agar and what is the use of agar in preparation of any media. So, nutrient media, we can say it is the balanced mixture of nutrients which was dissolved in water. So, what is the need of this nutrient media? They are most commonly used for cultivation of microorganisms in the laboratory. Now, by using different nutrient media, we can isolate, identify, and enumerate or count the number of bacteria which are present within particular sample. Now, while designing the medium for the cultivation or the isolation of the microorganism, we should design the media in such a way that it must supply all the necessary nutrients which are required for cellular growth as well as to maintain that growth of microorganisms. Necessary nutrients are required which was mainly supplied by nutrient medium. So, a suitable cultural medium, it must supply carbon and energy source. In addition to that, it must supply nutrients, growth factors, all the things which are required for the growth of that microorganism should be supplied. Now, some of the naturally occurring substances can also be used or it can serve as the excellent medium for the cultivation of bacteria. For example, milk. Milk will consist of all the nutrients which are required for the growth of that organism to be take place. Usually, skim milk are mainly used for the cultivation of bacteria. While using natural medium for the cultivation, we must dispense that natural material or the substances which you are using for the cultivation into the tubes or flasks, and you must make it sterilized before use. So media, either it is in the form of nutrient broth or nutrient agar, it should be designed and prepared so that it should have all the nutrients which was required for the growth of that organism to be take place. So nowadays commercially available dehydrated products are available. So by adding convenient amount of water, we can prepare the medium which was act as a excellent nutrient medium for the growth of microorganisms. Now the commercially available dehydrated media, they are available in powdered form. The preparation of any bacteriological media, it involves different steps. First step is each ingredient must be dissolved in appropriate volume of distilled water. Then determine the pH of medium by using either pH meter and make the necessary adjustment. If you want to prepare solid medium, then you must add desired amount of agar and boil the medium to dissolve the agar. Then dispense the medium into the tubes or flasks. The medium is sterilized by using autoclave. Some of the medium ingredients are heat labile and therefore for sterilization of such a kind of the ingredient you can go for the filtration method. Sterilization by filtration. Microorganisms require different micro as well as macronutrients for their growth. While designing the medium, it should support all the macronutrients which are required for the growth of microorganism. As we have discussed, the nutrition is the most important part of microbial physiology, as nutrition provide the nutrients which are required for the growth of that organism. The requirement of different organism is different with respect to the nutrients, as some of the organism they require some nutrients in larger quantity while some of the nutrients are required in just a trace element or just a trace amount. So those nutrients which are required in large amount they are called as the macronutrients, while those nutrients which are required in just trace amount they are known as micronutrients. Microbial nutrients are the compounds which are constructed from the chemical elements. So, as you know, some of the elements are required in larger amount. Examples are the hydrogen, oxygen, carbon, nitrogen, phosphorus, sulfur, selenium, and they are dominated. They are also known as the essential nutrients. In addition to this, 50 other elements are, although they are not required, but they can be metabolized by microorganisms. Now let's discuss the common media constituent for bacterial growth. So, to provide nitrogen source or the amino element, we can make the use of peptone, protein hydrolysate, infusions and extract. To supplement the growth factor, we can make the use of blood, serum, yeast extract or vitamins or NAD, nicotinamide adenine dinucleotide. Energy sources can be supplemented by using sugar, alcohol and carbohydrates. Buffer salts can be supplemented by using the sources like the phosphate, acetates and the citrate. Mineral salts and the metals can be supplied by using metals like the phosphate, sulfate, magnesium, calcium and iron. So, for the selection of a particular kind of organism, we can make the use of selective agents. So for that we can make the use of either chemicals or different antimicrobials which are produced by that organism. In addition to that, we can also make the use of different types of the dyes. Now, next one is the indicator dyes. These are also the constituent of media, as they can be used for the detection of acid or the alkalis which are produced by that particular bacteria. So we can make the use of chemical indicators like the phenol red or the neutral red. Now, in addition to that, the gelling agent that which can act as a solidifying agent also, we can make the use of either agar, gelatin, alginate, silica gel. or if we have added these sources within the medium, then the media will become solid. As you know, these microorganisms, they require carbon and the nitrogen, which are serve as a macronutrients. So these they can make the use of different organic compound as a sole source of carbon. While heterotrophic bacteria, they can, by using this organic compound and by using them, they can make their new cell material. So for that, they can make the use of variety of organic compounds like the amino acids, fatty acids, organic acids, sugars, nitrogen bases, aromatic compounds, and different organic compounds can be transported from the environment to the microbial cell, which can be catabolized, and that by using that organic compound they can withdraw energy, as well as by breakdown of that complex material they can synthesize their own new cell material. While in case of the autotrophic organism, they can make the use of CO2, and by obtaining the energy either from light or variety of inorganic chemicals, they can build their cellular structures. As you know, the bacterial cell is consists of about 13% of nitrogen. Now, why the cells require nitrogen? This nitrogen can be required in the synthesis of proteins, nucleic acid, and several other cell constituents. So, bulk amount of nitrogen is available, which in the nature is in inorganic form like the ammonia, nitrate, or the nitrogen gas. Now, in addition to this macronutrients, carbon and nitrogen, the cells may require different macronutrients. So let's have a look. But these nutrients or the elements, they require in smaller amount. Phosphorus. Phosphorus is a key element in synthesis of nucleic acid and phospholipids. Now, this phosphorus, it is mainly supplied to a cell as phosphate. Next, a nutrient is the sulfur. Sulfur, as you know, there are only two amino acids which consist of sulfur, that is the cysteine and the methionine. Now, for the synthesis of this cysteine and the methionine amino acid, sulfur is required. In addition to that, it is also involved in several synthesis of several vitamins like the thiamine, biotin, and the lipoic acid. Sulfur, it can also be supplied to cells in several forms, including sulfide, or we can also make the use of sulfate. Next one is the potassium. Potassium is required for the activity of different enzymes. Next source is the magnesium. Magnesiums, they used to stabilize the ribosomes, which are mainly involved in the protein synthesis process, in stabilization of the membrane also, and nucleic acid. These magnesium ions, they play important role. In addition to that, for the activity of certain enzymes, magnesium is required. Next one is the calcium. Now, calcium as is mainly required by the endosporulating cells, that is those cells which have a tendency to form the sporulating structure that require calcium in larger quantity. So this calcium, it helps to stabilize the microbial cell wall, and it also play a role in the heat stability of endospores. In addition to calcium, sodium is also required by different organisms, and the level or the quantity of sodium requirement will be differ. Some of the bacteria which are present within the sea water, they have a tendency to tolerate high levels of sodium, and therefore they require more quantity of sodium for their growth. So such a salt-loving organism are known as halophiles. If we compare the freshwater species, the freshwater species, they are able to go in absence of sodium, potassium, magnesium, calcium, and because these are mainly supplied to the cells as a salt, typically as chloride or the sulfate salts. Now next source or the next constituent is the peptone. As we have discussed, it is a complex mixture of partially digested protein. So from what are the sources of the peptone? So peptones can be obtained from meat, heart muscle, casein, fibrin or the soybean. Now peptone is the most important constituent of amino acids proteases in organic salt, which includes phosphates, potassium, magnesium and growth factor, including nicotinic acid and riboflavin. Now, the peptone is mainly supplies nitrogenous material. In addition to that, peptone it also acts as a buffer. Now, in our laboratory, we mainly store this peptone in a tightly closed container as it is hygroscopic in nature, and if it exposes to water, it becomes sticky. Next one is the water. As you know, water is the most important dissolvent. So, in case of the culture media, to dissolve all the medium constituent or the component, we require water. Mainly, distilled water is preferred while preparation of the media. If you are making the use of tap water, then your tap water it may consist of different salt like the calcium and the magnesium salt. Now this calcium and the magnesium salt they can react with the phosphate which is mainly present within the peptone as well as phosphate is also present within the beef extract. So when this calcium and the magnesium salt it react with the phosphate, then it results in the formation of calcium phosphate or the magnesium phosphate salt. And these salts they are water insoluble, and therefore if we prepare the media by using tap water, precipitation kind of the reactions will take place. So this insoluble calcium phosphate as well as insoluble magnesium phosphate salt, once they are formed, they inhibit or prevent the growth of microorganisms. Next one is the salt. Salt are required or the presence of sodium chloride in the nutrient medium. It is required to maintain the salt concentration in the medium. As we have discussed, salt is also required to maintain the osmolarity. That is the osmolarity or the salt concentration of the medium. It must be similar to that of the cytoplasm of the microorganism. And therefore, we have to add appropriate quantity of sodium chloride in the nutrient media so we can maintain the salt concentration. Next one is the yeast extract. Yeast extract is mainly prepared from the cells of baker's yeast or the Saccharomyces. So, yeast extract, it consists of carbohydrate, amino acids, growth factor, mainly vitamin B group and inorganic salts. Now, this yeast extract is used mainly as a source of vitamins, and instead of if meat extracts are not available, then you can make the use of yeast extract. Next one is the meat extract. Meat extract is prepared from fresh meat by using hot water extraction process. Now, if you are making the use of meat extract, then it will serve or it will consist of source of gelatin, peptones, proteases, amino acids, creatine, creatinine, purines, mineral salts, carbohydrates, growth factors like thymine, nicotinic acid, riboflavin, pyridoxine, and pantothenic acid. Next one is the agar. Agar is the most important chemical constituent while preparation of the solid media. Agar is a long-chain polysaccharide which is mainly obtained from the seaweed algae which are belong to the class Rhodophyta. So this algae, the algae from where we have extracted agar, they are called as agarophytes. It mainly includes Gelidium, Gracilaria, Hyphae, Gelidiella, Ceramium. These are the some of the examples of algae. So, if we say agar, then it is a mixture of two polysaccharides like the agarose and agaropectin. The concentration of agarose in agar is 70% and the concentration of agaropectin is 30%. In addition to this, it also contains calcium, chloride, magnesium, sulfate, iron, etc. Now, agar or the agar-agar powder, it is most commonly used for the preparation of solid media. Now, what are the different properties of agar? So, first one is we can prepare the agar from different seaweed algae. Agar, if we are making the use of 2% of agar, then it can act as a good solidifying agent. Agar does not have any nutritional value, but it can be used as a solidifying agent. These are the references. Thank you.